for our final session here on that's going to be on ensuring efficient and consistent high-quality generic drug development. Okay, so please join me in welcoming our presenters. First, we'll hear from Dr. Greg Huang, a senior chemist in the Division of Product Quality Assessment, or DPQA 9, that's within the Office of Product Quality Assessment, or OPQA. And then we'll hear from Dr. Feng Liu, a lead pharmacologist in the Division of Bioequivalence 1 within the Office of Bioequivalence in the Office of Generic Drugs. Then we'll hear from Dr. Priyanka Ghosh, a lead pharmacologist within the Division of Therapeutic Performance 1 in the Office of Research and Standards within the Office of Generic Drugs, followed by Dr. Lei Zhang, Deputy Director of the Office of Research and Standards within the Office of Generic Drugs. And then we'll also hear from Dr. Yuqing Gong, a senior pharmacologist within the Division of Quantitative Methods and Modeling, or DQMM, and that's also within the Office of Research and Standards within the Office of Generic Drugs. So now I'll hand it over to our first presenter, Dr. Greg Huang.
Hi, I'm going to talk about the guidance for industry content and the format of composition statement and the corresponding statement of ingredients in the labeling in NDAs and ANDAs. Even though it is about a policy, today I'm more focusing on the CMC perspective regarding the ANDA and the NDA submissions, what should be adequate information to be included in a composition and the labeling. So today's learning objective, you will be able to describe content and the format in the composition statement of a drug product. You'll be able to describe what exactly to include in the statement of ingredients in the labeling. And you will be better understand the requirement for consistency in composition information through the NDA and the ANDA submissions. So the FDC Act and 21 CFR 314, they are requirements for NDA and ANDA to provide full list of components used in the drug product manufacturing, and also provide a clear statement of composition in the drug product. So this guidance is a draft guidance. It is fairly new, published 5 months ago in April, first draft. So this guidance recommends what kind of accurate and complete information regarding the composition statement and the statement of labeling. Ingredients in labeling should be provided. This will ensure that FDA approved labeling and composition contain adequate qualitative and quantitative information. And this definitely will help a CMC assessment to ensure we have an efficient assessment process and minimize the communication cycles and lead to fast approval of and NDAs. So what exactly we mean by composition statement? So it should include the qualitative and quantitative information of active and inactive ingredients of a drug product. Specifically in the ECTD submission is in module 3, section P1. And what is the statement of ingredients? It means the information of all the ingredients in active and active listed in the labeling, which is in the ECTD module 14. and including other types of FDA-approved labeling, such as the cartoon container labeling, patient insert labeling. So what should be included in a composition statement? So first of all, first is the name of all the active and inactive ingredients with clear identification, such as grade and type. So for this, you can refer to the established name of USP NF monograph and also the FDA Global Substance Registration System, or GSRS, to refer to these source. And for the GSRS, the ingredients has a unique ingredient identifier, or called a Unicode. So it also, second component of the statement should include the reference, compendial reference standard. If it's a USP NF, then it should be clarified. If it's in-house, then it's in-house. And also the primary function of inactive ingredients, which means the role of the ingredients played in the formulation. You can further refer to the USP General Chapter 1059 for the excipient performance for the functional categories of the excipients for further information. It should be a straightforward terminology, such as preservatives, antioxidants, buffering agent, or salt-forming agent, etc. should be clear. So the fourth component is the quantity of each ingredient. It includes the concentration in the liquid, mg per mil. Mg per mil, and also the amount or strength per unit. And also it can be the percentage by volume or weight. And it should be in the same units for each quantity category. So here's an example of the composition statement. So this is an example for injectable injection drug product. Sodium drugazide is a hypothetical API for the purpose of a presentation. So it includes name. So the name is very clear and specific. Either it's a API sodium salt, or it's a sodium citrate dihydrate, or a citric acid monohydrate. Edited sodium dihydrate is very clear. Okay. Second component is about the material related to the compendial standard. So in this case, they are all USP compendial. And the function is API, tonicity, buffer agent, preservative, and the quantity in mg per mil, mg per vial unit, and the percentage of weight by volume. And this is continued example for the name. It's very clear, specific, povidone K17. And also the compendial standards, it's could be in-house and USP or national formulary or in-house. Yeah, the function and the quantity. So this is a complete and accurate representation of the formulation. So I'm going to give a little bit more specifics how to identify ingredients in composition statement. First of all, the active ingredients, it can be in a salt form, even though the potency in the labeling typically is expressed in the potency of the free base or active moiety. So, both of the information should be clarified in the composition statement. And if API is a mixture, so it can be a chemical isomer or sterile isomer, then it should be stated clearly, and the composition. If it's a racemic mixture, it also should be clarified. So for inactive ingredients, hydration form, the grade composition of the ingredient, and a type of polymer. It can be molecular weight or the different composition. We are going to give a more example in later slides. And also purity. So I'm going to give an example to identify API in the module 3, P1. P1 is the description and the composition of drug product. So here it says sodium jugoxide is a salt of jugoxide converted by reacting with sodium hydroxide. Each ml of the drug product contains 27.3 mg sodium jugoxide, which is equivalent to jugoxide, which is a free base of 25 mg. So in this case, we assume assume the API is a mixture. So you would say jugoxide is a mixture of jugoxide A and jugoxide B in a ratio of 3:1. This is an example to explain it. So yeah, this is very clear. So about the inactive ingredients, what information to include? First of all is the hydration form. So in this case, the sodium citrate, citric acid, added sodium. So they are differentiated by anhydrous, monohydrate, or dihydrate, other forms. I know the USP monograph may cover different hydration forms. So, but the recommendation is to be clear what form you are using in the formulation. So the next one is a polymer type like povidone. The same USP monograph can cover different nominal K values. And the second example is hypromellose. Differentiated by substitution type of methoxy and hydroxy propoxy percentage. The next one is hydroxyethyl cellulose, which contain different proportion of hydroxy ethoxy group. And the carboma has a different type by the cross-linking and copolymer composition. And more example about the composition, like a stearic acid, it can be differentiated by the percentage of stearic acid and palmitic acid. And also the next example is about the purity content, acetic acid and glacial acetic acid. They're differentiated by the different purity, and alcohol and dehydrated alcohol, also the same, differentiated by the purity. So more specific about the statement of ingredients in the labeling. So this means the list of ingredients in module, ECTD module 1, section 14, it should be consistent with the statement of composition in module 3, P1. And qualitative information for all the active and inactive ingredients in the labeling, and a quantitative information of API for all drug products, and a quantitative information for inactive ingredients for parenteral injection. So here is an example. Again, it's for a hypothetical injection drug product. So the description of labeling in ECTD 1.14, it says each milliliter contains a drug dose, jugoxide 25 mg, equivalent to the sodium salt 27.3 mg, and the following ingredients: citric acid monohydrate 1 milligram, etc. So they are specific in the type of the ingredients and the amount, and also the pH adjuster. It does not, if it's a use adjuster, it does not have to be quantitative information provided, but definitely it should be listed in the labeling. So some observations during our CMC review, and this first one is observation. Second is how it should be clarified. So first, a compendial reference not identified. So it should be compendial or in-house. And hydration form not identified. Very common for like sodium, added sodium, and just added sodium. We don't know what kind of form it is. And sodium carbonate monohydrate, so it is clear on the right column. And the pH adjusting agent not listed or clarified. It's not in the labeling, or it can be missing from the composition statement also. So it should be clearly listed. And inconsistent unit of concentration for inactive ingredients. So sometimes all the ingredients are expressed in a percentage weight by volume, but one of them may be presented in a different unit. This is not a good situation, so it should be corrected. And for the conversion, you may also provide additional information such as density and the temperature, etc., to convert to a consistent unit of ingredients. Okay, so another example is a composition of active ingredients deviated from the UST monograph. So in the case, the raw material may be pre-processed. So in this case, is a pre-acidified polysorbate 80. So it's considered in-house, even though your starting material, polysorbate 80, is a USP NF grade, but this is pre-processed. And the grade of the inactive ingredients are not clarified. So on the right column, the clear example is a povidone 17 or hypromellose 1822. They are very clearly specified. So we just talked about the composition statement, how we should include in terms of content format, and also the labeling, what should be included in the statement of ingredients. Throughout the CMC submission, actually, there are more to be taken care of. First of all, is in the batch formula in the module 3P, section P3, and a master batch record, executive batch records. In the relevant modules, they should be consistent. Sometimes we see inconsistency, and this should be noted. So also, if the multiple manufacturing facilities include, there may be some variation. Okay, so the composition statement and the labeling should include all possible ingredients and levels in the composition statement, and be consistent in the labeling. So here's a few examples for inconsistency we observe in the NDA and the ANDA submissions. First example is acid base added as pH adjusters. But they are omitted either in the P1 composition of the drug product, or they could be omitted in the batch formula. So we needed to make sure that the composition table in P1 and the batch formula include consistent information about the pH adjuster. So the second example is a composition table lists the ingredient in fixed amount. However, in the batch record, it says QS. Okay, this is not consistent. So they should be consistently presenting each active and inactive ingredients and their amount. So with all that, I want to present a challenge question. First is the following should be included in the composition statement of a drug product. First, name of all the active ingredients and inactive ingredients. B, reference quality standard for all ingredients. C, primary function of active ingredients. D, quantitative information. And E, all. Which one? Oh, I heard all. I think all of you. Great. Okay. Second question, which is not needed in the statement of ingredients in labeling? This could be more than one answer, not sure. First, name of all active and inactive ingredients. And the pH adjusters, grade or type of inactive ingredients, polymorphic form of API, processing aid used in the drug product and manufacturing. Which one? Which one, D? D and E, okay, great. Yeah, so I should emphasize this is still the very first draft. Is still revising, and we are addressing public comments. So we definitely welcome more questions in the panel discussion. Actually, my colleague Rachel is a panelist, so we can discuss, and also refer to the 1059 for excipient performance as a resource. So in summary, composition statement should include clearly identified ingredients and their quantitative amounts. Statement of ingredients in labeling should list complete ingredients that are consistent with composition statement. And batch formula and manufacturing batch records in the ECTD submission should be consistent with composition statement. So I'd like to acknowledge my colleagues at Work Unit OPQ2 of OPQ for their feedback, and also our wonderful guidance working group when I prepared this talk. I got very good feedback, and I thank all of them. So next, I will invite Dr. Fang Lu to the podium.
Good afternoon, everyone. I will share with you the analysis of the first cycle Bioequivalence rate and major BE deficiency encountered from bioequivalence perspective. Self-introduction. I'm a team leader from Division Bioequivalence 1, OGD. Learning by sharing. When I prepare this presentation, two questions pop up in my mind. Number one, what is the first cycle Bioequivalence rate? Number two, how we can improve first cycle Bioequivalence? To answer those two critical questions, I will present the information as outlined here. Number one, purpose. Number two, data-driven outcome. Number three, tips that could improve first cycle Bioequivalence. Number four, summary. The more review cycle during the review process, the longer time to achieve Bioequivalence. Therefore, we'd like to share with you first cycle Bioequivalence and the major BE deficiency type encountered, along with those suggestions how to avoid those deficiencies. We retrospectively assess the submission quality and commonly observed BE deficiency from 729 NDAs submitted during GDUFA III Year 1 and identify 82 NDAs are BE inadequate. One review cycle is defined as FDA received the application to the first CR letter with either BE inadequate or BE adequate outcomes. The first cycle Bioequivalence rate is 87% for 335 NDAs assessed by OB. Out of 82 BE inadequate NDAs, 57 and 43% were classified as a BE major or minor deficiency, respectively. All right, it's a busy slide. However, we'd like to present the BE major deficiency themes from an overview perspective and summarize as a 7+1 top themes as in this graphic. Most of the deficiency themes are inadequate in vitro or in vivo B studies and method validation as well. We also identify the impact from other disciplines, include data integrity issue, identified by oath inspection, or the batch not acceptable as determined by OPQ, or exhibiting with a safety concern by FarmTalk. We will further explain the major B deficiency by using case scenario with tips how to avoid these pitfalls. For example, in case number 1, the statistical result provided by applicant only include 2 decimal places. Well, as our BE assessor found that the study failed when conducted confirmatory calculation applying 4 decimal places as a specific recommend in guidance. Please note that all in vivo B study and in vitro dissolution testing data are considered invalid once the biobatch has not been found acceptable due to many reasons, such as the API source has a CGMP issue, or drug products quality or stability issue. We found one case that the capsule stick together became too yellowish during the storage, so the batch is not acceptable. So all of the study considered invalid. So please see the tips here. Wisely select API suppliers. Ensure your drug product's quality and stability, and maintain the manufacturer site compliance. In this table, we summarize each BE major deficiency themes, followed by sample deficiency with the tips. For example, when exclude a subject data under product deviation, we suggest to submit a full investigation report from both bioanalytical or clinical deviation perspective. The key point is here with the real-time evidence, including your statistical plan. For those easily correctable deficiency, we suggest that before submission, carefully check if BE study report or data are complete and consistent as possible. For example, double check if your SAS data set submit as a CDX format. In many cases, you can avoid easy correctable deficiency. PSG is a useful resource to avoid those BE deficiencies. For example, original PSG include only 1 in vivo options. However, the revised PSG including both of in vitro and in vivo options. So you have more options. For those complex drug products with a challenging issue, we encourage you to discuss with us on your BE strategy before submission by appropriate approach. Of course, you may also propose alternative in vitro or any in vivo approach with a scientific justification. So here we have some so-called challenge question, but I think simple question here. Number one, the first cycle B adequacy rate. The answer is simple, C. So this a little bit challenging. In which of the following scenario could a biobatch still be considered acceptable? I better give you the answer quickly, okay? Age batch with appropriate stability, the rest of unacceptable. So in summary, the data-driven outcome indicate that first cycle B adequacy rate is about 87% for 635 NDAs assessed by OB. Please consider those tips in slides number A to 12, not K to 12, in the presentation. Let's work together. For example, communicate with us by appropriate approach on your BE strategy to bring effective, safe genetic drug products to American people. I appreciate my OB colleagues, especially Utpal, Tao, Chicha, Sri, Zen, Bing, and Pasa, for their help on this presentation. Of course, I appreciate all OB assessors and project managers. Without them, the data cannot be collected. Thank you. Let me turn this stage to Priyanka.
Okay. I'm going to continue the discussions related to ANDA approval and major deficiencies. And I'm going to use a case study with topical products for the purposes of this presentation today. So the learning objectives for us is to understand the ANDA landscape for topical products applied to the skin, specifically what kind of approaches are being utilized to support an evaluation of BE, and then to understand what the challenges are and how do we resolve those challenges so we can eventually get to first cycle approvals. So the outline of my presentation has a brief recap of the BE approaches that we currently recommend within product-specific guidances for topical products. And then the second part of the presentation talks about landscape analysis of ANDAs that have been submitted to the agency since GDUFA II, so FY18. And then, of course, the challenges and opportunities. So a brief recap from the presentation that Dr. Kelchen provided yesterday morning. There are predominantly 4 types of BE studies that are currently recommended within the guidances for topical products. We have the comparative clinical endpoint BE studies. These can be used by all products irrespective of the formulation. Then we have the vasoconstrictor studies. These are typically used by corticosteroid products. We have situations where a waiver of in vivo BE studies may be applicable, typically for topical solutions where bioavailability may be self-evident. And lastly, the characterization-based approaches that have been developed within the scope of the GDUFA Regulatory Science and Research Program. So let's now look at some data. So I'm going to take some time on the slide to orient you to the slide because all of my subsequent slides are structured the same way. This is a summary of the submitted ANDAs that we have received since FY18. And the way the data is presented here is data for a given fiscal year is normalized to the total number of ANDAs received in that fiscal year. For the purposes of this presentation, approved ANDAs indicate any ANDA that has been approved or tentatively approved. Pending ANDAs indicate any ANDA that is in pending status, either in a CR status with the applicant or in pending status with the agency, and of course, withdrawn ANDAs. So overall, if we look at all of the data that we have since FY18 till FY24, the data is as of June 30th of 2024, we see about 55 to 57% of the ANDAs that we have received during this time for topical products applied to the skin have already been approved. Now let's take a look at what kind of approaches were used to support BE in these submitted ANDAs. So if we focus on the left of the screen here, we see the characterization-based approaches. And since the publication of the guidances related to these characterization-based approaches in the FY 18-19 timeframe, we have seen a reasonable increase in the number of ANDAs that have utilized characterization-based approaches to support BE. And the number that I would like for you to keep in mind is this FY 2021 number, where about 70% of the ANDAs used the characterization-based approach. We have also seen consistent submissions for products which have used clinical endpoint BE studies, VC studies when appropriate, and of course waivers when appropriate. Now let's look at what has been approved. So when we look at approvals, if we focus on the characterization-based approach first, remember the number 70% that I mentioned for FY21, we see that about 35 to 40% of the ANDAs have been already approved. So overall, we see approximately 40% of the ANDAs that have used characterization-based approaches have been approved until now. We also see consistent approvals for the clinical endpoint and the VC studies, but I'm going to talk about those in a little bit more detail when I talk about pending ANDAs. If we take a closer look at the approaches that used characterization-based approaches, we see that a substantial number of those products have successfully completed IVRT studies, and also some of those products have successfully completed IVPT studies. So I just wanted to note that before we look at the challenges we face in this area. So now looking at pending ANDAs. As we look at pending ANDAs in trend with the submissions where we saw an increase in utilization of the characterization-based approaches, we also see an increase in the number of pending ANDAs when a characterization-based approach has been used, and I'll delve into this a little bit more on subsequent slides. For the clinical endpoint studies, VC studies, and waivers, we do not see as many pending ANDAs, but I'll talk about overall some of the challenges that we do observe. So for the overall pending ANDAs, if we were to break them down about the challenges, we see about 50% of these products still have outstanding challenges related to bioequivalence, pharmaceutical equivalence, and in some cases also labeling, for example. But the other 50% of the ANDAs where bioequivalence have been found to be adequate, there are outstanding challenges related to pharmaceutical quality. They could be related to either drug substance or drug product, and also related to manufacturing and process and the facility. A limited number of cases where we have labeling challenges. So one thing that I wanted to note here is, while the pre-NDAs that I have been involved in have predominantly focused on the BE approaches, because this is new in the area of topical products, there may be some room for discussing pharmaceutical quality-related challenges in the pre-NDA space when those are appropriate as well, so that we can resolve those challenges and get to that first cycle approval. Delving down a little bit into these pending ANDAs that utilize characterization-based approach, we see about a quarter of these ANDAs, quarter to 30% of these ANDAs have adequate IVRT study. For the remaining ones, while this segment looks big, I will note that the majority of the challenges that we observe with IVRT are primarily related to IVRT method validation or analytical method validation. And with the resources we have currently available, guidance, trainings, I think a majority of those challenges are resolvable within one cycle. With IVPT, however, we see that approximately 79% of the ANDAs that we have pending and which require an IVPT, the IVPT is inadequate. So this is a significant challenge for us. And when it comes to IVPT, we have observed that there are challenges related to method development. And what I mean by that is we observe that there is inadequate optimization, for example, with respect to the receptor solution. It is extremely important during IVPT method development to optimize the receptor solution for a given drug molecule, a given drug product, to ensure that there is sufficient sensitivity, sufficient solubility and stability of the molecule in receptor solution. And it is often not appropriate to just use a receptor solution that is available in the lab and proceed to method validation and pivotal studies. Another example of a challenge that we observe is with sampling frequency. We often see methods that are being submitted where a standard sampling frequency is utilized without any consideration for how that sampling frequency impacts the shape of that permeation profile, and if that sampling frequency allows us to truly compare two products. So one potential option to resolve some of these challenges, because we do recognize there are challenges with IVPT method development, is to potentially discuss with us as you are developing your IVPT method, as you are running into these challenges through the pre-NDA path, and specifically through the product development meetings. The other major challenge that we observe related to IVPT is related to method validation. And there we see challenges related to the selection of the dose, for example, for the IVPT sensitivity study, or the selection of the alternative formulation for the IVPT selectivity study. And in this situation also, we feel like we have guidance out there about how to select the dose. We have presentations out there about how to select a dose that will give you the separation of the profile between your target dose and your selected dose to be able to establish the sensitivity of the IVPT method. But we also recognize that since assessing sensitivity and selectivity is a qualitative assessment at the moment for our guidances, this can lead to some challenges. So again, as you are developing your IVPT method, as you are validating your IVPT method before you proceed to that expensive pivotal IVPT study, if you have questions, we strongly encourage you to interact with us through either the product development program, or in some situations, even the pre-submission program may be appropriate. There are, of course, other issues related to aberrant data in IVPT studies. And given all of these challenges that we are observing, we hope to host a CRCG workshop in March of next year to discuss a lot of these challenges in a lot more detail. So in conclusion, I would like to say that the landscape analysis of the ANDAs in the topical product generally indicate that characterization-based approaches have been predominantly used to support ANDA submissions since GDUFA II. And overall, not just using characterization-based approaches, we have approved over 175 topical ANDAs since FY18. Among the pending ANDAs, there are challenges related to pharmaceutical quality, labeling, and/or BE. However, for the products where characterization-based BE approaches were used, the predominant challenge appears to be related to the IVPT studies. So we would like to, again, recommend engagement with agency early during product development, as you are developing your methods, as you are validating your methods, so we can resolve some of these challenges and support product approval. So with that, my challenge question: what are the pathways that can be used to interact with agency? A product development meeting, a pre-submission meeting, a post-complete response letter related meeting, controlled correspondences. I would say you can use all of the above, whichever seems most appropriate for your specific question. So with that, I would like to acknowledge my entire team within the Office of Research and Standards, and I would like to invite Dr. Lei Zhang for her presentation related to ICH M13.
Okay, good afternoon. Next, I'm going to give an update on the ICH M13A, which is the first ICH guideline for bioequivalence. And you may be aware from yesterday's opening remarks from Commissioner or from the news, this really represents the milestone for the generic drugs in terms of BE in the global level. So I will provide an overview of the M13 guideline series, not just M13A, but what's the plan, and also provide an overview of the final M13A guidance and major changes from the draft, and also what the major difference from the FDA current general guidance, as well as provide an update on the FDA planning on the implementation of M13A for the generic drug development. So for this audience, you all aware the bioequivalence assessment is really very important for the establishment of the therapeutic equivalents for generic drugs to their respective reference list drug. And also you may be aware the reference list drug is the terminology FDA is using, but for the M13A, comparator product was utilized. Is equivalent to FDA's RLD, which represent the standards, this is the terminology we harmonize at the global level to represent the reference standard other regulated agencies will recognize for their regulation needs. And we also know BE is not only for generic drugs, they also be utilized by new drugs for their decision-making, either pre-approval or sometimes the post-marketing, post-approval formulation or manufacture changes. So it's very important a concept. However, traditionally, we know in the ICH level, there's no harmonized guidance for bioequivalence or for generic drugs. So this is really the first topic bring to the ICH. Starting with the reflection paper that was adopted by ICH in 2018 on further opportunities for harmonization of standards for generic drugs, which laid out a strategic approach on how we harmonize and provide these scientific technical standards for BE, starting from a more simpler product to more complex products. And the ICH also formed the generic drug discussion group following the reflection paper. The discussion group really have representations from not just the regulatory agency and the ICH, but also the industry groups to recommend topics for harmonization and what should be the order for prioritization. And they also supported the submission of the first new topic proposal, which is lead to the M13 expert work group formation, focus on the immediate release oral solid dosage form. And the M13 expert working group was formed since 2020 and mainly focused on immediate release solid oral dosage form. And based on the scope of the work, the EWG proposed to develop M13 in 3 series. So M13A really represents the first guideline under this series, focusing on the BE study design and analysis for immediate release solid oral dosage form. And later there will be a M13B is second in the series, will cover the topics on additional strength as well as including the additional strength waiver. And the M13C will cover the data analysis and BE for highly variable drugs, therapeutic drugs, as well as some more complex study design and data analysis, including adaptive design. The reason to break this into 3 series, so the working group will be able to break down the topics so we can deliver those finalized guidance in a timely manner, in sequential, but also consider there could be some overlapping while we work on those guidances in order to bring the efficiency. So right now the M13A is finalized. We are very happy. Now it's reached the step 5 and it's ready for the global regulatory agency to implement this guidance in the generic drug development. M13B is ongoing, and the working groups currently work on reaching consensus on the draft technical guidance to anticipate to reach step 1 towards the end of this year. And the M13C has not started yet. It will start after M13B is the draft is published and during the public consultation stage. So it is really a long journey when I'm looking back. I have the privilege to serve as the rapporteur for the M13 expert working group. And Lufa Temple, who will be on the panel, she is the FDA topic lead. We have a very diligent and collaborative global working group under ICH with 22 members, observers, as well as among them, 14 of them are the regulatory agencies. So we have been working very hard in the past 4 years by published this guideline, follow the ICH process for harmonization. Really, there are 5 steps as laid out in this slide. And we delivered the draft guidance towards the end of 2022, and we received a lot of public comments, and we reviewed all of those comments and the finalized guidelines based on the comments received and understand the areas that may need additional clarification. And the final guidance was finished and adopted by the ICH in July of this year. And now it's ready for, at step 5, ready for implementation. And the global regulatory agency under ICH also have their own plan for the implementation, and the FDA is also in the process of revise our general guidance as well as the product-specific guidance to align with M13A. So the overall content of M13A in the final guidance does not change from the draft guideline. There's 5 sessions, including the introduction that will cover the objectives, background, and scope. And the key content is in the section 2, which talks about general principles for establishing BE with both study design and the data analysis part. And we focus on non-replicated study design. And we also have 4 sections covers various specific topics on endogenous compounds, other immediate release dosage forms, which including the oral disintegrate tablets, chewable tablets, and oral suspension, as well as the fixed dose combination. And we also have the session talk about the pH dependency, when you may need to consider one additional study under the with a acid-reducing agent in order to establish BE. And we also have a section on documentation and glossary. So during the public comment set period, we received close to 800 non-replicate comments, and the majority of them are related to Section 2. It's not surprising because this is the key part of the guidance. We have 77% comments related to that section. In addition, we have 10% of comments also related to Section 3. And the major comment themes are focusing on, because we introduced the risk-based approach for determining whether we need 1B studies or 2B studies under what fasting or fed conditions. So there's a lot of clarification needed on how you define high risk and when fed B study is needed. We also received some other comments related to how you define the dose proportionality. This is also related to M13B in terms of additional strength waiver, and also study population, data analysis, various aspects of it, group effect, and also alignment of study design with product labeling. So the expert working group review all the comments, and the modification are being made to based on those comments received. And also, in addition, we also realized some topics may need additional clarification or rationale, which we don't have the space in the guidance to provide that. So when we finalized the guidance, the working group also developed a questions and answer document. It's published the same time as the final guidance. So we don't think there's a major changes from the in terms of the recommendations from the draft guidance, but we did make modifications on several sections, mainly to provide clarification to help the implementation or make them more practical for the regular agency to implement the guidance. So in particular, the high-risk product, what does the risk mean in this under the context of M13A? It is really to mean the risk of the B inequivalence or B failure for those products which has special characteristics. They may result in a high risk of the B inequivalence under the fed condition due to the food effect, differential food effect caused by formulation or manufacturing process. And also the data analysis part has some more clarification provided, as well as PK dose proportionality assessment, early exposure. We, in addition to the partial UC, we also introduced the Tmax may be used, and also additional clarity on pH dependency when you may need an additional study, and what justification you may provide. So as I mentioned, the working group provide additional questions and answers document to help provide further clarity and also assist the implementation. As you can see, all the Q&A are focusing on section 2, 3, and 4. In addition, as part of the ICH process, a step 4 presentation was published the same time. This is serving as the basis for training materials to help the implementation, and the links on the slide. So if you read the guidance, before the M13A, the different agencies do have, even the B has been used for many years. The regulatory agency globally do have different recommendations for B study design and the criteria to support the generic drug approval. This may result in inefficient drug development, increased cost and time. Additionally, maybe cause some drugs may not be approved in certain market. And the major difference we are aware is the FDA generally recommended both fasting and fed studies. However, several other regulatory agencies, including the European Medicines Agency, EMA, they generally recommend one study, which is fasting studies. So those differences in the general B guidance can lead to the different product-specific guidance recommendations, which both FDA and EMA has PSGs developed in addition to the general guidance. So this table just really to show you those differences in the general guidance does translate to the PSG recommendation. If you look at the comparison between US FDA and the EMA, in terms of PSG, although FDA do develop more PSGs than the EMA, but if you look at the percentage-wise, we do have a majority, like 86% PSG prior to M13A recommend both fast and fast studies, while EMA only have 16%. And they have majority recommend the fasting studies only, because this is reflected by the general B guidance differences. As we are aware, the M13A introduced the risk-based approach to determine the need for the B study condition. So for high-risk product, as I briefly alluded to, those are the products which difference in the formulation or manufacture process may not be detected with a single B study, either in the fasting or fat study. Fat condition may not be extrapolated to predict B under other conditions. Therefore, the guidance recommend the B study for those type of product, B should be conducted in both fast and fed conditions. And also Q&A touched further rationale on why we recommend this. And for non-high-risk products, on the other hand, the results from a single study, which typically could be a fasting study, could be extrapolated to predict fed B study outcome or vice versa. So the B test only one condition is sufficient. And the guidance, the revision, you can see there's some slightly different language used in the final guidance, but the concept is not really changed, but we just try to articulate better how you identify a product as a high risk, non-high risk, what you may consider when you develop your product to consider whether you need to do 2 studies versus 1 studies. So really we need to look at the reference product or ILDs, drug substance characteristics, coupled with their formulation, design, or manufacture process, and what's the labeling used, what's the food effect, how they those with relation to food, and their specific product formulation that consider high risk, which a couple of examples were given in the guidance, which include the solid dispersions, microemulsions, lipid-based formulation, nanotechnology, and some other specialized technology. And also in the guidance also alluded that the test product also need to consider how you, similar to the reference product, just because some product or some reference product may not deemed to be high risk. However, although some product may not use the complex formulation, but the specific characteristics in the formulation, if it's different between the test and the reference product, they can result in a formulation that may modulate the food effect different, and they might be there might be a need to conduct a study under both conditions or provide justification. And also in the guidance, both in the draft and also in the final guidance, talk about the meal type for Fed B studies. So for high-risk product, the guidance recommend in respective of the product labeling, the study need to conduct both fast and fat, and the fat, it will be the high-fat, high-calorie meal. However, for the non-high-risk product, if the Fed B study is needed, especially when that's where the labeling indicates the drug should be taken with the food for a PK reason, then either a high-fat, high-calorie meal or low-fat, low-calorie meal can be used, administered, and also labeling. And also if the labeling specifies certain meal, that could be the meal used for the B studies. and also the additional rationale we'll provide in the Q&A. So this kind of slide summarize the current FDA B guidance versus the M13A in terms of fast and fed B studies. So FDA gonna change from the current recommendation, which is for pretty much for almost all oral IR drug products, asking both fast and fed B studies will move towards the risk-based approach as delineated in the M13A. And as I already mentioned, so for the high-risk product, probably both fast and fed study, but for non-high-risk product, they could be either fasting-only study or fed-only study, or either fasting or fed. Depends on the situation as delineated in the guidance. So FDA is really leading the harmonization efforts, and ICH recognize the importance of the harmonization in bringing the access to the medicine globally. And because of the M13A not being finalized and adopted, and FDA will revise majority of the IR oral products, PSGs, to align with M13A, following the risk-based approach, by considering the BCS classification of the drug substances, formulation design of the ROD, and what's the food effect, what's the labeling condition, whether the drug is to be taken with food or without regard to food. So based on this assessment, we look at more than 1,100 PSGs, current existing PSG, which covers those products. And we recognize that more than 800 of these TSGs can be determined as non-high risk. So we will recommend from the current 2B studies to 1B studies, either in the fasting conditions or fed conditions, which means we'll remove either fed B study or fast B studies based on the labeling condition. And you can see we published the forecast list. There's a link down there last Friday, so you will see what are those 800 plus PSGs are. And we also aim to publish the revised PSG in the first quarter of 2024, which target in October next month. So the significance of M13A is very obvious. So this is the first harmonized ICH guideline focused on BE. It will impact a significant portion of the pharmaceutical market. This is also the reason why the ICH started IR product first, because it's almost occupy about 50% of the market or approved new drugs in the US. And it will reduce the need for additional in vivo B studies due to the divergent recommendations. And it will support a more streamlined global drug development. Although I did not really cover much, is the M13 also recommend a study design where multiple arms can be utilized in the study design to include the multiple reference products or comparative product or multiple test product in order to improve the efficiency and satisfy the multiple jurisdiction requirement. And this will benefit the patients by increasing access to generic drugs. So here are the key takeaways. So M13A has been finalized, and it's adopted by ICH, and it will be implemented or is in the middle of implementation for ICH regulatory members, including FDA and other regulatory agencies. It will facilitate the regulatory approval, promote confidence in the quality of the generic drugs, and also enhance global access to those medicines. FDA is updating the relevant guidance, not just the PSG, but our general guidance will also be updated to align with M13A, with relate to the IR part. And also just remember, both FDA's current guidance and ICH M13A do offer flexibility. The ANDA applicants may provide appropriate scientific justification if they decide to take an alternative B approach, different from the guidance, deviate from guidance. That is also possible. So today I only can have a very high-level update on this important guidance, but we plan to have a SBIA webinar on this guidance on November 21st, save the date, for 2-hour webinar, which will have additional detailed detailed update on the key scientific thinking behind the recommendations, FDA implementation plan, and address your questions more thoroughly. Now is the challenge question. Challenge question number 1. Following the implementation of ICHM 13A guidance, which of the following will represent the most significant changes in the FDA recommendations for B studies for immediate release solid oral dosage drug products? A, B studies designed with regard to food conditions, B studies population, C, B for long half-life drugs, D, batch size for pivotal B studies. So the correct answer is A, B study design with regard to food conditions. Challenge question number 2, FDA will revise product-specific guidances for immediate release oral drug products to align with ICHM 13A. Is true or false? I hope you get it is true. Thanks. So here are the resources. Hope we'll provide you additional information. And I would like to end that we, as OGD, as the FDA, we collaborate globally to resolve scientific and regulatory challenges to ensure access to generic drugs. With that, I'm going to turn over to Yu-Chin, who will provide an update on the MIE program, pilot program. Thanks.
Hi, I'll be providing a talk on the Model Integrated Evidence, the MIE Industry Meeting Pilot Program for Generic Drugs for the first year review. This is my disclaimer. So in today's talk, I'll provide an overview of the Model Integrated Evidence, the MIE Industry Meeting Pilot Program, and share what we have learned through the first year of the MIE Meeting Program, and recognize the potential topics for discussion through the MIE Meeting Pilot Program, and then learn how to prepare an effective MIE Meeting request package. So in OGD, there are 3 major areas to apply the quantitative methods and modeling. They are mechanistic models, including the physiologically based PK, the PBPK, and the CFD models, like we had some talk yesterday and today. This can be applied to both the oral drug and non-oral drugs. And the second main area is the pharmacometrics and the quantitative clinical pharmacology, which includes the population-based model, the pharmacokinetics, and pharmacodynamics models. And the third area is the data analytics, which I put here as big data, including the machine learning toolsets, analytics for complex mixtures, and risk-based model, and etc. OGD launched the MIE pilot program on October 1st, 2023. So the MIE pilot program aims to offer the generic drug company a meeting opportunity who intends to submit an MIE approach in their ANDA. And the MIE program is open for the whole life cycle of the ANDA, including the pre-ANDA and ANDA stages. So the pilot program allows enhanced scientific communications on a broad range of quantitative methods and modeling techniques to address generic drug development issues or questions that are either out of the scope or cannot be sufficiently addressed by the existing pre-ANDA and ANDA scientific meetings. For example, you can discuss the common modeling issues across multiple products and common modeling approaches for non-complex products in the MIE program, which are not the main focus in the current pre-ANDA and ANDA scientific meetings. And the main objective of the MIE program is to provide a dedicated regulatory platform for interactions on MIE to foster early and focused interactions between industry and FDA on MIE approaches for establishing bioequivalence in generic drug development. And you may also refer to the general principle documents link in the bottom. In the MIE meeting, the following scenarios may be granted to discuss the innovative MIE-focused approaches for B establishment that cannot be effectively addressed under the existing GDUFA scientific meetings, and to discuss the non-complex products with complex modeling approaches, and to discuss the novel data analytics tools and approaches, for example, the machine learning and artificial intelligence for BE establishment and assessment. Here I'm providing a brief overview of the MIE pilot program timeline. So first I'd like to note the MIE pilot meeting is not a GDUFA meeting, so it doesn't follow GDUFA goals. However, we're following approximately the timeline that listed here. So the MIE pilot meeting includes 3 stages. In stage 1, the applicant submits an MIE meeting request. Within about 14 days, FDA will assess and evaluate the meeting package, and at its sole discretion, FDA will make the decision to grant or deny the meeting. If the meeting is granted, in stage 2, it may include up to 2 meetings with the applicant. So within the 30 days of the grant date, FDA may schedule and host an optional orientation meeting with the applicant where the applicant will present its MIE approach in a video conference and have a direct communication with FDA regarding the details of the MIE package. And whether, regardless if the orientation meeting is scheduled or not, the applicant will have the final meeting with FDA in 120 days to 150 days, depends on the orientation video conference meeting. And FDA will issue the preliminary response 5 days before the meeting days. And the stage 3 is the post-meeting communications. Approximately 30 days after the final meeting, FDA will send the official meeting minutes. Here I'm providing some case examples through the first-year program experience to share with you what is opportunities of this MIE meeting program. So the first case example discussed a complex regulatory issue where modeling and simulation were used to characterize PK differences between available and discontinued products, providing a scientific bridge for BE demonstration under the circumstance where neither the R&D nor the RS is accessible. So in this case, we discussed feasible model-based bridging between the 2 oral dosage forms, where the applicant will be able to conduct a comparative in vivo PK study for its test product compared with an available product, although this product is not R&D. However, the model-based bridging helped to identify the PK differences between all these products and eventually provide a BE evaluation for the applicant's test product versus the R&D. The MIE meeting provided in-depth technical discussion regarding the data source and the bridging strategy and the modeling techniques. So through this MIE meeting, a feasible model-based bridging approach was established. And I'd like to note that this is a case example where the MIE meeting program allowed to discuss the modeling approaches for non-complex drug. In my second case example, the applicant submitted the MIE meeting request to discuss an alternative MIE-focused approach for BE establishment for a long-acting injectable. So this long-acting injectable, the bioequivalence study needs to be conducted in patient population to reduce the recruiting burden for patients. So the applicant submitted a mechanistic PBPK modeling approach informed by in vitro characterization data and a small-scale in vivo validation study. In this package, here are some example topics that were discussed in the meeting, including the detailed technical discussion regarding modeling methods and modeling validation strategy, and in vitro experimental methodologies for PBPK model development and validation purpose, and the approach for virtual bioequivalence analysis, for example, the selection of the sample size, the power of the study, and the variability, and risk assessment, including type 1 and type 2 error analysis. So since launch, 2 meetings were granted and completed the MIE process. The MIE pilot meeting provided a unique forum for FDA and industry scientists to gain a shared understanding on model development, model performance, and the use of modeling to address a specific issue in generic drug development. So in those meetings, FDA's participation are multidisciplinary, and it provides inputs from the ORS DQMM-led multidisciplinary review team. And the MIE meeting allows alignment in MIE strategy across FDA review team and with the applicant at early stage. And through those meetings, we found an orientation meeting was helpful to understand the proposal at early stages and enhance the mutual understanding. So the video meeting format for both the orientation meeting and the final meeting was especially beneficial as it allows direct communication with the applicant on the complex modeling questions. Here are some examples for the good candidates for the MIE meetings. I'm putting here as 3 big categories, but there could be many, many examples on each categories. So first one is you may discuss innovative MIE-focused approaches for BE establishment to address complex issues for a single product or multiple products in an MIE meeting. Some examples are listed here. For example, like yesterday, we have a talk discuss a common strategy for validating a CFD model or platform towards predicting the regional deposition for oral inhaled drug products. So this common strategy can be discussed under an MIE pilot program. And another example is an alternative study designs for long-acting injectable supported by MIE, as my case example 2 here. You can also discuss the non-complex products with complex modeling approaches in an MIE meeting. For example, in my case number 1, the model-based bridging for alternative product when the R&D and RS is discontinued. And the other example could be the BCS-based biowaiver or the other study waivers for the oral drugs. You may also discuss the novel data analytic tools in the MIE meeting. For example, the equivalence analysis of complex particle size distribution, the new quantitative approach for API sameness assessment, and application of machine learning and artificial intelligence for bioequivalence establishment. And for more details, you may refer to the SBI 8 webinar in January 2024, where we have a dedicated webinar on the MIE pilot program. To prepare an effective MIE meeting request package, you should consider have a sufficiently developed scientific proposal where the meeting questions are relevant, critical, and clearly stated. And adequate documentation for the modeling approach can be very helpful to be more effective in the communication. For example, the purpose of the MIE approaches and how it will be used to address the question of interest and inform regulatory decision-making, and sufficient details for underlying assumptions and the model building process, and a clear model validation and verification strategy, including how you will be using the data and the future data to support, and risk analysis and assessment, and modeling files and supporting data sets. So you may submit one single request for MIE along with a complete meeting package to FDA at mie@fda.hhs.gov. And a confirmation of meeting submission recipient by FDA will be sent via email that will indicate FDA received date. Here are my challenge questions. Which are the following scenarios may be granted as an MIE pilot meeting? A, innovative MIE-focused approach for BE establishment. B, non-complex products with complex modeling approaches. C, novel data analytic tools and approaches for BE establishment and assessment. D, all of above. So the answer is D, all of above. Challenge question 2, which of the following statement is not true? A, within 14 days of the meeting package recipient date, FDA will strive to evaluate and assess the meeting package submission. FDA, at its sole discretion, will determine if an MIE meeting request will be granted or denied. B, the MIE pilot meeting is part of GDUFA meetings and will follow GDUFA meeting process and timeline. C, during the initial assessment of the MIE meeting package, FDA may determine and schedule and host an optional orientation meeting within the 30 days of the grant decision date. D, regardless if an option orientation meeting is scheduled, the applicant will participate in a final meeting via video conference with FDA. So B is not true, as the MIE meeting is not part of the GDUFA meetings. Here are some take-home message. So the pilot program serves as a dedicated regulatory platform for industry to explore the proposed MIE approaches, obtain FDA's advice on their feasibility and advancement, and address relevant scientific and technical questions. And I'd like to note the MIE meeting program is open for the whole life cycle of the ANDA, including pre-ANDA stage and ANDA stage. And the MIE pilot program is ongoing to accept more meeting requests so that areas of improvement can be identified to support future recommendations. So we highly encourage applicants to participate and engage with the FDA early in your product development program. Here are some resources that might be helpful. The first link is to the general principle guide documents for the pilot program. And the second link is to the January 2024 SBIA workshop, where we had a dedicated webinar to discuss the MIE industry meeting pilot program. And a closing thought, FDA has been openly encouraging the use of quantitative methods and modeling approaches with MIE to support the development and approval of generic drug products. So consider the MIE pilot program when requesting the meeting with FDA. Now I'd like to invite all the other speakers and panelists to join me to the panel. Thank you.
All right, thank you again to our presenters for their excellent presentations, and we will now have our final question and answer panel to include all our presenters, and also joining the panel, we have Dr. Nilofer Tempal, Associate Director for Scientific Quality with the Office of Bioequivalence in the Office of Generic Drugs, as well as as well as Rachel Erdman, JD, Regulatory Counsel with the Division of Policy Development with the Office of Generic Drug Policy within the Office of Generic Drugs as well. All right, and as before, attendees in the room, you are invited up to line up at the mics to ask any questions. And for those online, if you haven't had a chance to submit your questions using the on-screen link and QR code, please do so now, and we'll answer as many questions as time allows. All right. So again, in the room, this is the last chance to ask the experts while you have them here. So at any point, please feel free to come up. And in the time being, we'll go ahead and start with our online questions then.
All right, so our first question is for Dr. Greg Huang. So for lyophilized product, if RLD composition doesn't provide salt form quantity, but only includes free base quantity per vial, as an ANDA holder, do we have to specify the salt form quantity in composition?
Yes, both the salt form and the free base concentration and the name should be included in the composition table. And also it should be clarified in the labeling.
Thank you for that response. And now we have a question in room.
So the question is for Dr. Huang as well. So is excipient function part of the Q1, Q2 evaluation? So for example, if we match in all the other chemical characteristics as well as the quantity, however, we list the different primary function as the RLD holder, would that make us not Q1 equivalent?
This is an assessment question. Basically, the terminology may be some different in the naming, but it should not affect the Q1, Q2. Q1, Q2 is based on the qualitative and quantitative information, but we understand that some different applicants may use slightly different naming, but the role should be equivalent. And
for example, like BHT, we list as antioxidant, they list as preservative, but this is an extreme case since they're both exceptional. But in that situation, they're quite different in terms of their functions. Would that be okay?
Yeah, you might get a question if we are not clear about your description of the role of the excipient. Yeah, you might get a question in the CMC review.
Okay.
Thank you.
Thank you for that response. We'll go to another online question.
All right, so we have another follow-up question for Dr. Huang. Should all inactive ingredients be included in the outer pack or container label?
Yeah, all the ingredients should be included in the labeling, the name, the list, the full list.
Thank you for clarifying that. We'll go for another online question.
All right, we have another question here for Rachel Erdman. So should applicants round the amounts in the statement of ingredients in the labeling?
Well, thank you. Generally, no, we don't request that you round the numbers. The number of significant figures should match what's in your composition statement. Thanks.
Thank you for that response too. We'll go ahead and continue with our online questions.
All right, this question is for Dr. Fong Lu. If OPQ identifies GMP issues related to an API supplier, but the applicant showed that the API batches do not have concerns related to GMP issues, will FDA accept the biostudy?
It's case-by-case scenario. Semi-justification we can consider.
Thank you. And we still don't have any takers in the room quite yet. Oh, okay, actually we do. All right. So thank you for coming up, and please go ahead, ask your question.
This was a great program. This is a hypothetical question to anybody in the panel or for the entire thing over here. I've always seen that, okay, brand drugs, and now we are talking about generics. So in case the brand manufacturer decides to make the first generic, does it mean that he has to just do a label swap? Like, does he have to do an ANDA with just a labeling requirement, or what happens there? You know, if it's the brand, like let's say you can take, I don't know if it's Mylan or Teva, for the EpiPen, they made the generic epinephrine injection, right? Was there an ANDA process for that, or was it just, like any of the products, like let's say any of the monoclonal antibodies or any of these really complex drugs, like do you have to do another ANDA process even though it's the same manufacturer who's got the brand approved?
Okay, so I'll try to.
Kind of, that's why I didn't want to ask anything, but it's the question that's been intriguing me the whole 2 days, you know, like, because there is a commercial value in having a generic product because you don't want to lose the market. So if you decide to, because it's going to expire, it's going to, so I don't know what's the FDA take on that.
Okay, so what you are referring to is really the authorized generics. That's what we call as the authorized generics. So you, as the innovator, when you want to market this as an authorized generic, all you're doing is just changing the labeling in the front. Then there is no ANDA submission. There is not that I'm aware. I wouldn't say there is no ANDA submission for that. But if you are marketing an authorized generic, you have to, I'm not an expert here in this authorized generic field, but in terms of you have to inform or there is a authorized generic site, you get an NDC number and those things. So you may have to just look into the FDA's, some website for unauthorized generics for additional information. Thank you.
All right. What a Great example. If anyone else here has some questions they've been wondering about, now's a great time. We'll do another question here, actually, yeah?
Hello. Yeah, I have a similar type of question. So, some of these drugs which have been approved long back, ages back, now as you get a new drug or generic drug product out, you go through a full characterization, and there's a whole bunch of specifications, including impurities and stuff like that. And as part of the ICH guidance, with the impurities, you sort of show some of those things. What if the brand themselves have those impurities which are higher than the ICH guidance? What happens in those cases? And it was not in the specification because it's approved ages back.
I think maybe this is a little out of scope of this particular session, and I'm not sure if anybody else wants to answer, but I think this is a little out of scope on this ICH and impurities.
Sure. All right. Well, thank you, Sil, for being willing to ask and that response. And actually, we'll move on to an online question first. Yep.
All right, this question is directed towards Dr. Priyanka Ghosh. The first thing they said was, thank you for providing a very informative presentation. Regarding the characterization-based BE approach, emphasis on the IVPT and IVRT aspect for establishing the BE of topical products, will there be a bioequivalence data summary table that is formatted and tailored for this type of submission, as the current model bioequivalence data summary tables seem to gear towards clinical data? Thank you.
So at the moment, we do have some summary tables available for IVPT and IVRT that are accessible through the FDA's website. However, if you run into challenges as you are trying to utilize them, I would encourage you to let us know so we understand where the challenges exist and we can update them so they are easier to use.
Thank you for that response. And we had another in-house question.
Hi, thank you for coming out today. My question is about the ICH M13 and harmonization. So first, we thank the FDA for driving and leading some of that harmonization, all those efforts. We understand the challenges of that. Where I work at AAM, we work with one of our partners, IGBA, the International Generics and Biosimilar Association, and we advocate on certain issues. One of the issues we talk about is single global development. And with that, we think about three pillars. We think about the legal frameworks in different jurisdictions. We think about harmonization at BE, which you talked about. We also think about single global development or single global comparator. On slide 76, you mentioned multiple comparator, multiple regions. Can you give us some thoughts or some ideas of how we move the concept of single global comparator down the line, or how we push that concept a little further, so we're not doing multiple comparators in multiple regions for the same product? I hope that's not out of scope.
Okay, we'll get a start, and Lufa can always add on. Yeah, we heard you. We did hear that feedback. But however, in the guidance, we did talk about the selection or acceptance of the comparator product. It is really bound by the regional legal jurisdiction. So it's like ICH will not be able to harmonize that in terms of, which acceptance comparator will be accepted by which regulatory agency. So the solution we provide is really what you mentioned, is you could, in a single trial, you can include more than one comparator. And if you only rely on one for what, so the multiplicity, we also talk about multiplicity adjustment. In that case, you don't really need to adjust the multiplicity if you only plan to submit the set of data to one particular region for the one particular comparator. I don't know if that addressed your question, but that has been discussed, and we just don't have the bandwidth, I think, in ICH to harmonize that. Do you have any?
Yeah, it's more than the bandwidth. I would say that's just a guiding principle of ICH, that we harmonize on the bioequivalence, whatever standards, on the scientific and technical standards of whatever the expert working group is working on. while staying within the jurisdictions, or the regulatory framework of the different regions. So since every region, like EMA, USA, like, each one requires a very specific reference standard against which the study needs to be done, that's something which is dictated by the regulations, and so that's why it's out of the scope of ICH. So yeah, it's not ICH, maybe some other way to do it.
Please also consider comparator variability from scientific perspective.
All right, thank you all for that. We'll go to one more in-room question here.
I have a very simple question. Does the MIE pilot program cover only generic drug or new drug as well, or maybe drug device combination as well?
So I will start and Lei can add. The MIE pilot program currently covers generic drug. You're asking for drug device?
New drug and the drug device combination.
So in new drug, there is an MIDD program, which has similar function to discuss the modeling. So for the MIE, it's the one I'm showing, it's mainly focused on generic drug.
Thank you for that clarification. Now we'll do some online questions.
All right, so our next question is for Dr. Lei Zhang. So prior to the M13A, I was instructed by FDA to conduct both fasting and fed studies for my product, which I think is non-high risk based on M13A. Now M13A is under implementation. Do I need to submit a controlled correspondence asking the FDA to adjust the recommendation, or will it be sufficient to rely on the revised guidance and move?
Yeah, thank you for this question. Yeah, now we are in the middle of revising all the product-specific guidance. I would suggest look at the forecast list. If it is being listed as a being in the plan for revising, that's a hint about what direction FDA's current tentative thinking is. But you can also wait till the PSG is published. You can follow that. I do not think the person need to submit another control correspondences, but if they choose to, it's okay. But I don't think it's really necessary. They should follow the FDA guidance. Thank you. Updated guidance.
Any more comments from the panel? No? Okay, we'll move on to the next online question.
All right, this question is for Dr. Nilofer Tempal. So with this, what should a firm do if it is about to conduct studies under both fasting and fed studies based on previous PSG recommendations for a non-high-risk product? Should it obtain permission from the FDA to drop the fed study, or should it just do it on its own?
No, if you are planning to do 2 studies and if the revised guidance is already posted, which is recommending maybe 1 study, then that guidance is what you should generally be following. So is that the question? I'm sorry.
I believe so, yes.
Yes, okay. Yes, thank you.
All right. So we'll go ahead and do another online question.
Excellent. This question is directed to Dr. Feng Liu. So if applicant uses alternate API source because of CMC issues with the original API supplier and submits CMC data along with the comparative dissolution data during a pending ANDA, will it suffice requirements for ANDA approval, or does the applicant have to conduct BE studies with alternate API source batches?
Can you repeat this question?
Of course. If an applicant uses alternate API sources because of CMC issues with the original API supplier and submits CMC data along with the comparative dissolution data during a pending ANDA, will it suffice requirements for ANDA approval, or does applicant have to conduct BE study with alternate API source batches?
First, to figure out if the API source or supplier has any CGMP issue, I will refer you ask OPQ.
Yeah, we would suggest that you ask this in an appropriate response to FDA controls correspondence, since this is very application specific. Thank you.
Yeah, thank you for clarifying that. All right, and Again, we'll continue with our online questions, but still in the meantime, if anyone in-house has anything, feel free to come up. The invitation is still open to come to those mics. So we'll go ahead online.
All right, so this question is for Dr. Greg Wong. So if we have complex inactive in formula, is it okay to list only flavor name, or should we include all components of that flavor?
All components of the flavor and the quantitative information should be provided in module 3P1 composition, okay? But in the labeling, you may not need to provide that detail about the flavoring agent or coloring agent.
All right. Next question from online.
All right, so this one is for Dr. Yuchen Gong. Regarding MIE meetings, can we in industry use that to discuss modeling certain aspects of device design for a drug-device combination product? We invest a lot of resources into comparing these products, and we could use a modeling approach, including using virtual study subjects for comparative use human factors, for example.
Yes, the answer is yes. You can discuss those type of questions in the MIE.
Thank you for clarifying that. And so we'll keep on going with our online questions.
All right, so this question is for Priyanka. While I understand your presentation was limited to topical products and their respective draft guidances, they, however, list other dosage units that are not within the scope of these guidance documents. When working with IVRT and/or IVPT studies for these other dosage forms, for example, transdermal systems, is it recommended to apply as much as possible within these guidances to the IVPT study design, or are there additional guidances for these dosage forms expected in the future?
Okay. Yes, my presentation was focused on topical products applied to the skin, where we do recommend IVPT and IVRT studies within the scope of characterization-based BE approaches. When it comes to IVPT, and since transdermal dosage forms were mentioned in the question, I think we already do have a guidance, the product quality guidance for transdermal products, that has recommendations related to how IVPT studies can be conducted, for example, to support evaluation of heat effect of a generic transdermal product compared to the reference listed drug. In other situations, I think we currently do not recommend IVPT studies specifically to support product approval. So if you are wanting to conduct such studies, then I would encourage you to chat with us using the pre-NDA pathway.
Thank you for that response. We'll go ahead and go for another online question.
All right, this question is directed towards Rachel Erdman. FDA recommends listing the established name of an inactive ingredient in the statement of ingredients in a drug product's labeling. What if there is no official designated name under Section 508 of the FD&C Act?
So if you're saying that there's no official name and there's nothing in a compendium or anything like that, like USP, then you can generally use the common the common name of the drug, and of course just double check to make sure that that's acceptable. Thanks.
Thank you. Do we still have some more questions coming in online?
I can get some.
Okay.
So we have this next one is directed towards Dr. Fong Liu here. Can we consider the dissolution data from the comparative dissolution testing supporting the API source change as the same as that of in vitro dissolution testing for supporting waiver of in vitro BE study requirements for the lower strengths?
No, it's different. Number one, let me answer. Number one, for waiver of your lower strain, the study should be conducted with. Let me figure out. It's different with T versus R. For the other situation, like for API source change, this should the in vitro dissolution conducted with pre-change with a post-change of T. Those difference, T was R, pre-change T, post-change T. That's different.
Thank you for that. All right, and let's see, do we have, okay. Any other last call for questions here in the room? Again, this is the last chance, don't miss the opportunity. All right, well, in that case, we'll go ahead and thank our presenters again and panelists for all their helpful information. We'll give them another round of applause. Okay. And panelists, you can go ahead and take your seats, but everyone else, if you want to stay put just for something special, and that is our closing remarks from Dr. Robert Leinberger, who serves as the Director of the Office of Research and Standards, or ORS, in the Office of Generic Drugs, or OGD. Dr. Leinberger leads OGD's implementation of the GDUFA science and research commitments, including internal research activities and external research grants and collaborations to ensure the therapeutic equivalence of generic drug products. And ORS also provides pre-submission advice on complex generics through pre-ANDA meetings, product-specific guidances, and correspondence responses. So I'll hand it over now to Dr. Leinberger.
Thank you very much. I'm very excited to be here to close out this meeting. We had a fantastic workshop. Hopefully you all learned a lot about the science behind the generic program and some of the ways that it makes an extremely positive impact on our review processes, on the development of your products that you submit to FDA, and on access to generic versions for the American public. The generic program won't exist, especially for complex generics, without a strong scientific foundation. It's, if you didn't have that, then it's not clear, well, what studies do you have to do? You'd fall back to a situation where let's just repeat the studies that brand products did. And that pathway is not going to lead to efficient access to generic products. And I think you saw throughout this workshop the way that the scientific foundations of the generic program are being built up and lead to more efficient product access. On the first day, you saw looking at in vitro release methods for complex suspensions and the scientific work that helps you develop those approaches. You heard about topical products and the Q3 approaches and how they're taking over from the clinical endpoint studies and being the more appropriate pathway for products. You heard a session on inhalation products. Again, not very many generics for the inhalation products, but you saw the progress toward inhalation products based on PK and in vitro studies instead of clinical endpoint studies. We hope this will lead to a surge in interest in generic companies in developing inhalation products that meet the needs of the American public. Again, a huge area of opportunity for you in the generic industry created by our scientific work. And then you also heard about drug device combination products and some of the challenges in developing those types of complex generics. We don't have access to generic products unless you all in the industry submit applications. And I think you heard today in the early morning talks, a lot of examples of some of the things that go on during the review process for some of these complex ANDA products. And hopefully this gives you the insight as to how to make your submissions better, but also that you ought to be investing in development programs for these complex generics because you see that it's possible. You heard about long-acting injectables. I think 10 years ago, can I make a generic for that? It seems impossible. Now it's not just possible, it's done. And if you're not doing it, you're probably falling behind. Same thing with, you heard a talk on peptides, right? 10 years ago, oh, this is a recombinant source RLD. Generic's not possible. The only thing I can do is a B2 application. That's not true anymore. Again, another huge opportunity just in time for what we know is now the biggest part of the pharmaceutical market for generic competition to be active in that. If we didn't build the scientific foundations to change how we're thinking, then we wouldn't have the possibility of generic versions for all the GLP-1 agonists. So again, huge foundation. If you're in the generic industry, keeping up with these are how you plan and establish your business to be competitive in a new environment and provide the things that the American public wants. They want access to generic versions of all of these complex products. And the bigger the impact the products are, the more that they want and need those generic products. And this is, the huge value that you in the generic industry create. We at FDA recognize that, and we constantly try to develop processes, procedures, way to interact with us to make that process more efficient. You heard about our model-informed evidence meeting pilot, right, as well as other ways you can interact with us. And finally, today you heard about the M13 update. So the scientific force toward more efficient bioequivalence, not just limited to complex products. Even the commodity tablets and capsules, we continue to try to make that process much more efficient. And I think, look at what the impact of that is. Look at that forecast list that Lei mentioned. 800 PSGs tells you what study is going to be removed. I would think you're going to get a lot of questions about that, but this is, again, a huge restructuring. If you think about the benefit of that, that's probably 400 Fed BE studies that last year you would have submitted, next year you don't have to. That's, I don't know how much the studies, I know how much PK studies cost me, I don't know how much they cost you, but that's hundreds of millions of dollars in savings for the generic industry. And that's not just savings next year, right? That's savings sort of every year going forward that you're producing and maintaining these products that are essential to just the everyday use. I mean, they're commodity products, but commodity product is still making a huge public health impact. People take commodity products every day for blood pressure medications, cholesterol-lowering medications, huge public health impacts from high quality commodity products. And we continue to make that process easier and more straightforward to make those products and ensure the supply of that. So we hope that you take that money that you save on those BE studies and you can invest it into product quality, reliability, and ensuring continued access to these products. And with that, this is a fantastic opportunity for you all in the industry, in the room and online, to get in touch and hear about these innovative approaches and what's changing, so you can keep ahead of the, all of these things are going to have significant impacts on your businesses, business processes, and scientific developments going forward. This is a fantastic place to keep up with that. And I do want to thank the people that really made this possible, especially the scientific organizing committee. So we have one, I don't think when Lei is here, I want to recognize her as the chair of our scientific committee. I want to recognize Lei as our executive sponsor to keep everything working, and also recognize Maria as leading the project management activities on that. But we also had a large committee of scientific experts. You saw many of them as the panelists or the leads session moderators for today. Just recognizing a few of them, recognizing some of our project managers, Irfan and Karen, recognizing Jan, Andrew Babiskin, Deanna Vivian, Ki Ren, Susan Levine, Fallon Smalls, Sheila Rajesh, Ahmed Zidan, all of these people from OGD and OPQ who are members of our technical committee who really helped put together the programming, recruit the speakers, organize the topics, help develop, moderate the questions. So we had a great discussion. So I want to thank all of those people. So join me in thanking them. And with that, I'll turn it over to our SBIA staff to close out the meeting and tell you how to get all the final benefits from this meeting and everything you need.